Today we will learn the Chapter 9. Chapter 9 title is Histone Ubiquitylation. Chapter 9 is composed of three subjects starting from the histone H2B monoubiquitylation and also reversible histone ubiquitylation focused on the DUB and proteasome. Finally, we will cover H2A monoubiquitylation. So in this slide, I will explain the first subject, histone H2B ubiquitylation. So previously in Chapter 1 and Chapter 8, we briefly covered histone ubiquitylation, especially chemical modification in the histone tail, and also ubiquitination can be happens. And also in Chapter 8, we learned that H2AZ contains three ubiquitylatable lysine residue at the C-terminal. It's like this. So today's lecture we will cover histone H2B ubiquitylation. So we call it as the H2B UB1. Okay, so H2B UB1 was first identified in the Xenophus. So Xenophus, we call it as, in Korean, we call it as 아프리카 발톱 개구리. And also in East, okay, in East, especially the ubiquitination happens at 123rd location. There is the lysine, K is lysine. So especially in the East, this location, 123rd lysine is the ubiquitinated in the East. Okay, so this is the brief summary of histone H2B ubiquitylation. So in this slide, I will explain the histone ubiquitylating enzyme. So histone ubiquitylating enzyme is composed of the E1, E2, E3 proteins. Okay, let me start E1 proteins. So E1 is the ubiquitin activating enzyme. The very famous example is UBA proteins. The main function of the E1 is can activate ubiquitin using ATP. Okay, so this is the ubiquitin activating enzyme E1. So the main function is can activate, so this is ubiquitin, so can activate ubiquitin by using the ATP. So that's why they will make the ubiquitin E1 structure. Okay, so second enzyme is, we call it as the ubiquitin conjugating enzyme. The very famous example is the ROD6 proteins. Okay, so main function is the established or thioester linkage with the ubiquitin. Okay, so main function is to establish a thioester linkage with the ubiquitin. Okay, so this is the we call it as the thiol, and also this one we call it as ester. Okay, so thiol comes from the E2 enzyme, and the ester comes from the ubiquitin. So the E2 enzyme is established a thioester linkage with the ubiquitin. So this is the main function of the E2, and we call it as a ubiquitin conjugating enzyme. Okay, so the final one is, we call it as the ubiquitin ligase enzyme. The very famous example is the VRE1. Okay, so the main function is the recognized protein substrate. Okay, so this is E3 enzyme. First one is the recognized protein substrate. So this enzyme is recognized protein substrate. After that, and catalyzed the transfer of ubiquitin from ubiquitin E2 to protein substrate. Okay, so catalyze the transfer of ubiquitin from the ubiquitin E2 complex to protein substrate. Okay, so two protein substrate. So this protein, they have the, for example, this protein, they have the lysine at 123rd location. They have lysine. So E3 recognized protein substrate. So after that, E3 can catalyze the transfer of ubiquitin from the ubiquitin E2 to protein substrate. So that's why the H2B is ubiquitolated, especially at specific location. It's like this. In this slide and next slide, I will explain the histone trans crosstalk by H2B UB1. Okay, so trans crosstalk means that crosstalk happens between the H2B and the H3. Okay, so first of all, E1, E2, E3 induces monoubiquitylation especially in the H2B. So it will make H2B UB1. So E1, E2, E3 induces monoubiquitylation in the H2B and makes H2B UB1. So ubiquitin is consisting of 76 amino acids, creates gap between the nucleosome. Okay, so because the size is about 76 amino acids, so it functions as a wedge. So this is a tool. Okay, this is a wedge. Okay, so after we put the wedge into the substances, so it will make the space. Okay, so it means that ubiquitin function as the wedge. So it will create gap between the nucleosome. It's like this. Okay, so after that, step one is a histone methyl transferase. Okay, so especially step 1 is methylate the lysine residue in the H3. So methylate the lysine residue in H3 through the gap. Okay, so this is the histone trans crosstalk by H2B UB1. So we call it as this model as a wedge model. Okay. So next one is we also call it as the bridge model. Okay, I will explain it first. Okay, so E1, E2, E3 induces monoubiquitylation, especially in the H2B, and makes H2B UB1 is like this. So ubiquitin, consisting of the 76 amino acids, function as a bridge between the H2B and the H3. So it functions as a bridge between the H2B and the H3. So at that, step 1 is bind to the ubiquitin, and finally it will methylate the lysine residue in the H3. So once again, the ubiquitin consisting of the 76 amino acid function as a bridge between the H2B and H3. That's why we call it as a bridge model. Okay, so it means that trans crosstalk happens between the H2B and H3. Okay. So in this slide, I will explain the role of H2B-UB1 in transcription initiation. Okay, so in previous slide, we learned histone trans crosstalk by H2B-UB1. So there were two models. First one was the wedge model, and second one was the bridge model. So we don't know which model is right, but anyway, so histone trans crosstalk happens between the H2B and H3. So this crosstalk induces H3K4 methylation or H3K79 methylation. Okay, so these two marker is that this marker is a euchromatin associated marker. For example, H3K4 methylation, first methylation or second methylation and third methylation is also euchromatin associated marker. And also H3K79 3-methylation is also euchromatin associated marker. Okay, let me show you another example, heterochromatin associated marker. For example, the very famous heterochromatin associated marker is the H3K27 methylation. But on the contrary to the H3K27 methylation, these two methylation is the function as the euchromatin associated marker. Okay. So and this is also another example. So this is the H3K4, and also there is the first H3K4 methylation and the double methylation, and finally there is the H3K4 3-methylation. Okay, so in this H3K4 methylation and the H3K4 double methylation and H3K4 3-methylation is usually observed in the near the TSS transcription start site and also promote region, and also we also can find this the H3K4 methylation near the enhanced region. So it means that this methylation happens. So it means that it can make the euchromatin structure. So means that result in formation of the euchromatin structure. So after that, usually the RNA polymerase II can easily bind to the promote region. So it can initiate transcription. Okay, so this is the role of H2B-UB1 in transcription initiation. So let me summarize it again. So H2B-UB1 induces H3K4 methylation while H3K79 methylation. So this marker is the euchromatin associated marker. So resulting in the formation of a euchromatin structure. So it means that RNA polymerase II can easily bind to the promote region and also it can initiate transcription. So in this slide, I will explain the role of H2B-UB1 in transcription elongation. So especially the H2B-UB1 has the two functional roles during transcription elongations. So in this slide and next slide, I will cover the first role, and the following slide, I will cover second role. Okay. So this figure shows that the transcription elongation step. So this is the RNA polymerase II. So RNA polymerase II has the C-terminal region. Okay, C-terminal tail like this. Okay, so this is the PAF proteins polymerase associated factor. So this is PAF is function as the transcription elongation factor. Okay, so PAF is bind to the C-terminal region of RNA polymerase II. After that, PAF recruits Histone ubiquitylation enzyme, for example, RAD6 and BRE1. So RAD6 is function as the E2 and BRE1 is function as E3. So once again, the PAF recruit histone ubiquitylation enzyme or RAD6 and BRE1. So resulting in it can induce the monoubiquitination, especially in H2B. So they will make the H2B-UB1 is like this. Okay. So after that, H2B-UB1 can recruit another protein's FACT. So FACT is the histone chaperone. So this is FACT. So it plays an important role in doing the nucleosome disassembly and assembly. Okay, so this FACT protein also can selectively associate with H2A and H2B UB1 dimer. So this is the H2A, H2B dimer, but they have the ubiquitinated. So this is recorded as H2A, H2B, UB1 dimer. So FACT can selectively associate with this dimer and also can facilitate H2A, H2B, UB1 disassembly. So this is nucleosome. So the FACT is to facilitate H2A, H2B, UB1 disassembly from the nucleosome. Okay. So FACT has another function. They have the histone deubiquitination activity. So FACT can induce histone deubiquitination, especially in H2A, H2B, UB1 dimer. So it will remove the ubiquitination, especially in H2B, UB1. Okay. And also FACT has their main function is their function as a histone chaperone. So FACT can facilitate H2A, H2B reassembly into the nucleosome. Okay. So our conclusion is that during the transcription elongation step, so H2B is ubiquitinated. Okay, so that H2B UB1 can recruit FACT protein. So especially the FACT can selectively bind to the ubiquitinated H2A, H2B dimer, especially the H2A, H2B, UB1 dimer. So after that, it can facilitate histone disassembly and reassembly. Okay, so this step is which is the crucial for efficient transcription elongation. So once again, our conclusion is that H2B UB1 is a facilitate histone disassembly, so histone disassembly and the reassembly. So this step is which is crucial for the efficient transcription elongation. Okay. So in this slide, I will explain the second role of H2B UB1 in transcription elongation. Okay, so especially the RNA polymerase 2 has the C-terminal domain. Okay, so in the C-terminal domain has the consensus heptapeptide, which is composed of 7 amino acids starting from the tyrosine, serine, proline, threonine, and serine, proline, serine. And especially the second location, serine, and the fifth location serine can be phosphorylated. Okay, so this is the pre-initiation complex. Okay, RNA polymerase 2 is bind to the promote region. Okay, so at that time, RNA polymerase 2 don't have activity. Okay, at that time, transcription factor 2H has the helicase activity, so it unwinds the DNA structure. Transcription factor 2H also has the phosphorylation activity, so it will phosphorylate the fifth location of serine. So after that, transcription can be initiated. Okay, so during the transcription elongation step, especially the CTK1, this protein phosphorylate the second location serine 2 of heptapeptides. So CTK1 phosphorylate second location serine. Okay, so it will phosphorylate second location of serine. However, H2B UB1 inhibits CTK1 activity, so resulting in no phosphorylation. So resulting in the no phosphorylation in the serine second location of serine of heptapeptides. So it means that finally it will inhibit transcription elongation. So it will inhibit transcription elongation. Okay, so the first role of H2B-UB1 was it facilitate nucleosome disassembly and assembly. Finally, it facilitate transcription elongation. However, second role of H2B-UB1 is it inhibits CTK1 activity. So it means that resulting in no phosphorylation in second setting of heptapeptides. So finally, it will inhibit transcription elongation. So in this slide, I will explain the role of H2B-UB1 in chromatin dynamics. So to find the role of H2B-UB1 in chromatin dynamics, people make a nucleosome in which all H2Bs are substituted with H2B-UB1s. Okay, so after that, they compare the sedimentation coefficient of nucleosome containing H2B-UB1 with nucleosome containing H2B. Okay, so what is the sedimentation coefficient? Okay, before I explain the sedimentation coefficient, I will explain the principle of the sedimentation. Okay, so after the centrifugation, we will find that usually the more density molecules which has more density, so they will exhibit faster sedimentation. So they will this molecule will be precipitated very earlier. So like this. Okay, so what is the sedimentation coefficient? So actually the sedimentation coefficient is the ratio of particles sedimentation velocity. So this is a particles sedimentation velocity to the applied acceleration causing the sedimentation. So this A is the acceleration. But this is a very hard, very difficult to understand. So I will show you another example. So this is the graph, and y-axis is the density of a molecule, and the x-axis is the sedimentation coefficient of the molecule. Okay, let me start from the nuclei. So nuclei is the this is the cell. So this is the nucleus, and also we call it as a nuclei. Okay, so nuclei, they exhibit higher density, and also they exhibit higher sedimentation coefficient. How about DNA? So DNA is composed of the double helix structure like this. Okay, so they exhibit a medium density, and also they exhibit medium sedimentation coefficient. Okay, and finally there is also RNA. So RNA is a single-stranded molecule, is like this. So they show that lower density, and they show that lower sedimentation coefficient. Okay, so usually the ratio of the sedimentation coefficient is nearly highly correlated with the density, even though there is some exception here. Okay, so usually sedimentation coefficient is highly correlated with the density, even though there is some exception here. Okay. Okay, so after that, people perform the centrifugation. Okay, so this is the time of centrifugation. Okay, so initial step. Okay, see, this is the initial step of centrifugation. So molecules with a higher density, while molecules with the lower density, they are mixed together. Okay, so this is the intermediate stages. Okay, during the intermediate stages, you will find that molecules with higher density started to precipitate. Okay, and at the final step, you will see that molecules with a higher density is located, or molecules with a higher sedimentation coefficient is located in the bottom region, and also molecules with a lower density or molecules with a lower sedimentation coefficient is located in the middle region. Okay, and people found that nucleosome containing H2B-UB1, okay, they are located in the middle region. So nucleosome containing H2B-UB1 is located in the middle region. It means that nucleosome containing H2B-UB1 shows the lower sedimentation coefficient. However, nucleosome containing H2B, they are located in the bottom region. It means that nucleosome containing H2B shows the higher sedimentation coefficient. So it means that nucleosome containing H2B-UB1 has the more loose structure than the nucleosome containing H2B. So this is the experimental result to show the role of H2B-UB1 in chromatin dynamics. Okay, so in this slide, I will explain the second subject, reversible histone ubiquitylation. So I will explain specially focus on the DUB and the proteasome. So what is DUB? The full name of DUB is that the ubiquitylating enzyme. Okay, so now in this slide, I will explain the DUB for H2B-UB1. So what is the main function? The main function of DUB is to remove ubiquitins from H2B-UB1. So this is the H2B-UB1. So the main function of DUB is to remove. Okay, so to remove the remove ubiquitin from H2B-UB1, so it will make the H2B. So in yeast, the very famous the DUB in yeast is UBP8. So this is the subunit of the SAGAR. SAGAR is the histone acetyltransferase. So we will see some detail in the following the slide. And also in the mammalian, the very famous DUB is that USP3 and USP22 and the USP51. Okay, so table 9.2 summarizes the DUB enzymes. For example, the USP3, so USP3, the substrate for USP3 is that H2A and H2B. And also the substrate for USP22 is that H2A and H2B. And also the substrate for USP51 is also H2A and H2B. Okay, so the other USP is they only can remove the ubiquitin from H2A, not H2B. Okay. So in this slide, I will explain the 26S proteasome. So 26S proteasome is consist of the 19S, and also they have 20S. Finally, they have the 19S proteasome. So they are composed of three parts. First part is that 19S, and in the middle they have the 20S. Finally, they have 19S proteasome. Okay, so they have the 20S proteasome. They have a proteolytic function. So what is proteolytic function? So this function is that degradation of ubiquitylated proteins. Okay, for example, if protein is damaged, So this damaged protein can be recognized ubiquitin. Okay, after this ubiquitylated protein can be recognized by 26S proteasome. So finally, the damaged proteins can be degraded like this. So this is their main function. Their main function is proteolytic function. So they also have another function, non-proteolytic function. They function as molecular chaperones. So we will see each molecular chaperone function in the next slide. Okay. So in this slide, I will explain the 19S proteasome. Okay, so this is 19S proteasome structure, so which is composed of the lead part. Lead, we call it as 뚜껑, and also they are composed of the base part. So for example, if you see here, this is the 19S proteasome. They are composed of the lead part and they are composed of the base part. Especially in the base part, they have the 6 ATPase, which is composed of the RPT1 and RPT2 and RPT3 and RPT4, RPT5 and RPT6. So this protein can use ATP as an energy source. Okay, and also there is the temperature sensitive mutant pro ATPase. So this ATPase, so they made the temperature sensitive mutant pro ATPase. Especially they used the SOC125 mutant. So this is the ATPase gene structure, okay, which is composed of the 405 amino acid. Okay, so especially the SOC125 mutant. Okay, for example, 251 amino acid is the aspartate, but in this mutant, aspartate is changed to Y. Y is tyrosine. Okay, so this temperature sensitive mutant pro ATPase, especially the SOC125 mutant, so has ATPase gene variant. They have the ATPase gene variant that allows a normal function at lower temperature. However, they show that altered changed function, okay, mutant function, altered function at higher temperature. Okay, so people find that at higher temperature, so this temperature sensitive mutant pro ATPase, they exhibit no problem with proteolytic function. So it means that the main function, the proteolytic function is functional. However, they found that there is a problem with non-proteolytic function. So in previous slide, we mentioned that proteasome has a non-proteolytic function, especially the chaperone function. But we find that this mutant has the problem at this mutant, especially at higher temperature, they show the problem with non-proteolytic function. Okay, so we will see the specific non-proteolytic function of 19S proteasome in the next slide. So in this slide, I will explain the non-proteolytic function of 19S proteasome. Okay, so especially the H2B-UB1, so ubiquitinated H2B can recruit proteasome because proteasome can recognize the ubiquitinated proteins. So it means that H2B-UB1 can recruit proteasome. Proteasome is composed of 19S and 20S and 19S proteasome. Okay, so after that, we find that at higher temperature, the temperature sensitive mutants pro ATPase, especially the SOC125 mutant, shows the decreased H3K4 methylation and also decreased H3K79 methylation. Okay, what does it mean? And previously we learned that during the histone crosstalk, or during the histone crosstalk by H2B-UB1. So at that time we find that set 1. So set 1 can induce the H3K4 methylation or H3K79 methylation. So previously we learned that set 1 is involved in H3K4 methylation or H3K79 methylation. So it means that temperature sensitive mutant for ATPase, especially the SOC125 mutant, cannot recruit the set 1. Okay, so it means that we can find that this 19S proteasome is involved in the specific binding of set 1, the specific binding of set 1 to the chromatin. So this is the chaperone function. So once again, 19S proteasome has the non-proteolytic function. We call it as a chaperone function. The specific function is that specific binding of set 1 to the chromatin. Okay, so this is the non-proteolytic function of 19S proteasome. So in this slide, I will explain another non-proteolytic function of 19S proteasome. Okay, the name is that the regulation of the mRNA export by 19S proteasome. Okay, so this is proteasome. So proteasome can bind to the SAGA. So SAGA is that the full name of SAGA is SPT and the ADAR and the DCM5 acetyltransferase. The main function of SAGA is that it functions as histone acetyltransferase. Okay, so this binding is dissociate SGF73 and the DUBM. So this is the Ubiquitylation module. Okay, so this binding dissociate SGF73 and DUBM from the SAGA complex. Is like this. Okay, so after that dissociated SGF DUBM, so these proteins can interact with mRNA export machinery. Okay, so usually the apt RNA polymerase to synthesize the mRNA. So at that time, mRNA export machinery is bind to the mRNA. After that, so this the SGF73 and the DUBM and another mRNA machinery, they bind together. After that, it promotes mRNA export to the cytoplasm. Okay, so however, at higher temperature, the temperature sensitive mutants for ATPase, especially the SOC125 mutant, shows that decreased mRNA export. Okay, so it means that especially so this is the mutant form, especially the 19S proteasome may be bind to the SAGA. Okay, so but at mutant form, so 90S proteasome cannot bind the SAGA. So it means that there will be no dissociation of SGF73 and DUBM. It means that it cannot bind to the mRNA export machinery. It means that it will decrease the mRNA export is like this. So, okay, so in this slide, we briefly learn about another function of 19S proteasome in another function, especially the non-proteolytic function. So 19S proteasome plays important role in the regulation of mRNA export. So in this slide, I will explain the final subject H2A monoubiquitylation. So we call it as the H2A UB1. So H2A UB1, especially in the mammalian, so monoubiquitylation happens at K119th location of the H2A. So this is the H2A structure, especially at K lysine 119th residue can be ubiquitylated. Okay, so I will also explain the histone ubiquitylation enzyme for H2A. Okay, so histone ubiquitylation enzyme for H2A is nearly similar to histone ubiquitylation enzyme for H2B, except for E3. Okay, so let me explain the first. So histone ubiquitylation enzyme for H2A is composed of the E1, E2, and E3. First of all, E1 can activate ubiquitins using the ATP. After that, they will make the ubiquitin E1 complex. So after that, second enzyme, we call it as E2. So E2 is established a thiol ester linkage between the ubiquitin with the ubiquitin. Okay, so after that, E3 enzyme. This E3 enzyme is a recognized protein substrate, especially the H2A. After that, E3 is catalyzed the transfer of ubiquitins from ubiquitin E2 to protein substrate. So finally, they will make the H2A UB1. It's like this. Okay, so this procedure is exactly same as the H2B. However, the E3 enzyme is different from the H2B. Okay, in next slide, I will explain the E3 ligase enzyme for H2A. So in this slide, I will explain the E3 ligase for H2A. So previously, we learned that E3 ligase for H2B. So at that time, we learned that BRE1 protein is belong to E3 ligase for H2B. So in this slide, I will cover E3 ligase for H2A. So table 9.1 summarized the E3 ligase for H2A. Okay, let me start from the ring 1B. So ring 1B E3 ligase. Okay, so its substrate is the H2A, and also PRC1 complex is belong to the ring 1B. And also there are another complex is also belong to the ring 1B E3 ligase. Okay, so another E3 ligase for H2A is the 2A hub. So its substrate is also H2A. So anchor complex is also belonged to 2A hub. Okay. Okay, so in this slide, I will show you the detailed structure of each complex. So let me start ring 1B. So in ring 1B, PRC1 complex is belong to the ring 1B. So for example here, PRC1 complex is composed of the 4 proteins like this. Okay, how about FBXL10 and B-core complex? So they are composed of the several proteins like this. The final one is that E2F6.com-1 complex. And this complex is also composed of the several proteins. Okay, so also the 2A hub, there is only one complex. We call it as N-core complex. So N-core complex is composed of the several proteins like this. Okay, so in this slide, I will explain the histone trans crosstalk by H2A UB1. Okay, so let me explain the MLL3 first. So MLL3 is a histone methyl transferase. So it can methylate, especially the histone 3. Especially the histone 3 is composed of several amino acids, especially at post amino acid is the lysine, K is lysine. So especially the MLL3 can methylate H3K4. And also especially the H2A UB1. So this is the H2A UB1. So H2A UB1 inhibits the activity of the MLL3. So it means that resulting in the decreased H3K4 methylation. So this is the histone trans crosstalk by H2A UB1. So histone trans crosstalk between the H2A and the H3. So in this slide, I will explain the role of H2A UB1 in chromatin dynamics. Okay, so without link histone H1, okay, so this is the link histone H1. So without link histone H1, so this is nucleosome. So nucleosome has the open and flexible structure is like this. So they can be flexible is like this. Okay, however, H2A UB1, so H2A UB1 helps histone H1 bind to the nucleosome. Okay, especially the H2A UB1 helps histone H1, so this is histone H1 bind to the nucleosome. Okay, so it means that with histone H1, so nucleosome has more compact and rigid structure, consequently making the compact chromatin structure. So this is the role of the H2A UB1 in chromatin dynamics. So finally, the H2A UB1 is making the compact chromatin structure. So in this slide, I will summarize the role of monoubiquitination in chromatin dynamics. Okay, so first of all, H2B monoubiquitination. So H2B ubiquitination makes loose chromatin structure is like this. So make the loose chromatin structure. However, H2A monoubiquitination. So H2A monoubiquitination makes compact chromatin structure. So they have the opposite function. It's like this. So today we learned chapter 9. Chapter 9 title was histone ubiquitylation. First of all, we learned histone H2B monoubiquitylation. And secondly, we learned reversible histone ubiquitylation, especially focused on DUB and proteasome. So finally, we learned H2A monoubiquitination. Okay, so let me finish class today. Okay, bye.